talked a lot about starches. So when we talk about complex carbohydrates or starches, these are polysaccharides. They're going to be found in plants like potatoes, rice, and wheat. They're stored in the seeds, roots, and fruits of plants. And they're primarily composed of two different families of glucose polysaccharides or polymers. One is referred to as amylose. This is the linear alpha-1 4-linked glucose. And the other is amylopectin, that has those branch points. Okay, so similar to glycogen, it has these branching going on. So alpha-1 4-linked, these alpha-1 6-linked branches. And so typically, you'll find this type of percentage of amylose and amylopectin in a plant, about 20% amylose, the linear form, and about 80% of this branch. But there are some exceptions that will for example, waxy corn has a lot of amylopectin, and high amylose corn has higher amounts of amylose. But overall, this is generally a pretty good rule, that around 80% is present in amylopectin. So this is just another representation of these types of sugars. So amylose, you're going to get, is going to be linear, that alpha-1 4-linked, whereas amylopectin has these alpha-1 6-linked branch points. So it's going to have this kind of arrayed branching. This is a very good way to store glucose. We do that very similarly in our own bodies. So if you're eating animals or muscle or liver, you would be getting a similar type of carbohydrate in your diet from glycogen. And this is very similar to amylopectin. Both of them have linear alpha-1 4-linked regions with the alpha-1 6 branch points. And this type of formation is going to be really a good way to store glucose because you have a lot of ends where you can start chopping glucose off if you need it. Right? So say you need energy in your body, you can start taking a lot of glucose off. It's easy to mobilize. You can imagine if you're doing that with amylose and you're just taking the glucose off at the end here, it'd be a much more laborious process. Okay, so glycogen and amylopectin are very similar. Glycogen is more heavily branched. So these are estimate numbers, but it's just going to have more branch points than the amylopectin. So on this slide and the next slide, I'm just showing the structures for amylopectin and glycogen on this slide and the next slide for amylose, just so you can get an idea of what these look like. So here we have the cyclic form of our glucose, except it's in the chair conformation, which is a little bit more accurate for how these sugars actually look. And so if we look at one of these units, we have the 1 carbon, the anomeric carbon, and it's linked via a glycosidic bond. So we have the alpha-1 linked to the 4th carbon of the next glucose molecule, right? And if we work our way back, here we have the 4th carbon, so 1, 2, 3, 4 carbon, and it's linked via the glycosidic bond to the 1 carbon of the preceding glucose, and so on, right? So this is what we mean when we say a 1,4 linked glucose. Okay, and then we have a 1,6 linked glycosidic bond at the branch point. So again, we have that anomeric carbon, carbon 1 of glucose. It's linked via a glycosidic bond to carbon 6 of this glucose, right? And this gives rise to our branch. We have more 1,4 linked here. Does that make sense? And then for amylose, this would just be, so this will be the structure for amylopectin and for glycogen. For amylose, we'll just have the linear portion. Okay? And this is usually smaller, 100 to 1,000 glucose units, where this can be 1,000 to 1,000,000 glucose units present in glycogen and amylopectin granules. So this is just the linear portion of the 1,4 link. So it's also interesting to note that amylose is more resistant to digestion than amylopectin. And so this is because of that linear conformation and the lack of the branching. So the branching makes the amylopectin very able to dissolve in water, whereas amylose forms these hydrogen bonded structures. So it's forming hydrogen bonds with linear sections of amylose, and also with surrounding portions of water. That's what's shown up here. You can see that with our H2Os. So these would be two segments of amylose, and then they're layering in on top of each other in these packed hydrogen bonded structures. Okay? And so in raw potatoes is a really good example in which we get these tightly packed granules. So we just eat a raw potato. It's really difficult to, probably doesn't taste very good and it's hard to eat, but it's also really difficult for your body to digest that carbohydrate, the amylose that's present in that potato. Okay? And that's due to the hydrogen bonding and the packing of those amylose molecules. So in this case, cooking serves an important function in that it breaks up these intermolecular hydrogen bonds, and the starch then dissolves in the water. Okay, so cooking a potato not only makes it taste better, which is great, makes it easier to eat, but it also makes it easier to digest. Okay, so the heating is going to break up these structures and allow it to dissolve more in water, make it easier for your body to hydrolyze those bonds. So this would be an example of a resistant starch. Yeah. Yeah. So in this example, it's stacking. I think that was just kind of an illustration. It can form more three-dimensional helical forms, even when it's packed in together. So it could form more helical forms with these packing. Okay, and so I thought this was really interesting. So this idea that you have some starches that are more resistant to digestion and absorption, we've known about this for like a long time, right? And this is both in the case of eating some raw plants, or if you heat up a plant and then eat it, versus if you heat the plant and then allow it to cool, some of these structures reform, okay, when it cools. But this got like a huge amount of press last, this was March 2015, and these articles were just everywhere. And basically there was this presentation at a conference where someone had taken rice, they heated it, and then put it in the refrigerator and found that it absorbed less calories than the rice. So basically this is this idea that when you're cooling the rice, you're forming these more resistant structures as it cools and packs in. They're more resistant to digestion. So they're putting this out there as this idea that you can cut calories, rice calories, by cooking this in different ways. And this is definitely true. The way you cook or manipulate your food is going to influence how your body absorbs that food, how you digest that food, and the calorie intake. But I thought that this was a really interesting kind of idea, because most of the time when I think about food, and I think about the problems in the world, we're usually trying to increase crop yield and increase calories in crops and increase nutrient value in crops. But this is kind of the opposite, where we're trying to decrease the calories. So we have all these accessible calories, but now we're trying to make it so our body doesn't absorb those calories. It's kind of, where do you draw the line with that? Would we take advantage of this? Would we take a drug that would impair our digestion and absorption so we gain less weight? And those are out there right now for obesity treatments and for diabetics. So impairing glucose digestion and absorption. Would you take that as a weight loss strategy? And it seems like it's an interesting sort of thing to think about in terms of nutrition. So definitely the way you cook your food is going to influence digestion and absorption, mainly because of this formation of resistant starches. Okay, so in terms of digestion and carbohydrate hydrolysis, you have monosaccharides, so free glucose, free sucrose, free fructose. These don't need any more hydrolysis or breakdown. Your body's able to directly absorb simple sugars or monosaccharides. But in the case of disaccharides or polysaccharides, they're relatively large molecules, so you'll require hydrolysis or breaking of that glycosidic bond. So here we have sucrose. Glucose and join the fructose via that alpha-1,2 linked glycosidic bond. We have water coming in, allowing for us to hydrolyze and break those that disaccharide into two monosaccharides. And so our body is unable to absorb sucrose, but in the presence of an enzyme, sucrase, which mediates this hydrolysis, we break it down to glucose and fructose, two simple sugars, and then we can absorb those directly. So this is something to remember, only monosaccharides can be absorbed, so the objective is to enzymatically reduce sugar dimers and polymers to those monomers. Our body has specific enzymes which allow us to reduce these sugars into their monomeric subunits. But for some sugars we can't, right? Like for cellulose, that is a beta-1 form, we can't break that down, so we don't have that enzyme. So we're only able to utilize carbohydrates, so we have enzymes that allow us to break it down into those monosaccharides. Okay. And then other constituents may need to be digested first to release carbohydrates for digestion and absorption. And we'll come back to this idea. It's just the idea that these carbohydrates aren't alone in the gastrointestinal tract, but there could be, for example, granules of cellulose surrounding these carbohydrates, and that would need to be broken down first in order for your enzymes to access the sugars for further breakdown and absorption. Okay, so the constituents within the diet can influence your ability to digest and absorb these different carbohydrates. Okay, so carbohydrate digestion begins in the mouth. So as I mentioned previously, we secrete saliva into the mouth, and within that saliva there's an enzyme, salivary amylase. Right? And this is what initiates starch digestion. So the very first part of starch digestion begins in the mouth. Okay? So you have water in the saliva. This begins to solubilize sugars. And you have salivary amylase initially. So chewing is going to mix, of course, the food with the saliva and enzyme to help facilitate this. And if you ingest food really quickly, it's going to decrease the efficiency of salivary amylase during this chewing process. And salivary amylase can actually keep digesting carbohydrates as you swallow it, and will remain active in your stomach until it's mixed with the acidic environment of the stomach. So when you have that food bolus, initially, even in the stomach, it's going to remain active in the core until that's really mixed up into the chyme. And then salivary amylase will be inactivated at pH 2 or 3. Okay, so that's kind of said here. So the stomach doesn't secrete any enzymes that break down carbohydrates. If you recall, I mentioned chief cells secrete gastric lipase, breaks down some fat, and also secretes pepsinogen, which begins protein breakdown, but it doesn't secrete anything that breaks down carbohydrates. There will be a little bit of hydrolysis through salivary amylase coming from the saliva, but for the most part in the stomach, there's not a lot of carbohydrate digestion. It's mixing macromolecular breakdown and mixing into that acidic liquefied chyme. Right? So that low pH is going to stop any action of salivary amylase. So the majority of carbohydrate digestion occurs in the small intestine. So that salivary amylase starts the digestion, but it's a pretty minimal amount of digestion of carbohydrates. So the majority of this is accomplished in the small intestine. So there's pancreatic amylase. So this is the major catalyst of starch digestion. And so luminal carbohydrates are going to stimulate the transcription of this amylase, so this pancreatic amylase. So in response to carbohydrates in the duodenum, your body's going to start amping up production of amylase. Okay, so it actually stimulates transcription of amylase. And amylase, if you recall, is being secreted by the pancreas, right? From this acinar cells. So it's a soluble protein secreted into the duodenum from the acinar cells via the pancreatic duct. Okay? And it's stored in secretory granules in the pancreas. Okay? And this is just an example from our last lecture in which I showed you some images of these storage granules, secretory granules in a pancreatic acinar cell. And these are going to go exocytosis, right, which is that snare-mediated fusion of those vesicles with the plasma membrane so it can expel the soluble contents into the pancreatic duct. And so the secretion of these granules in response to food is an example of regulated secretion. talked about this constitutive secretion. This would be an example of regulated secretion. Okay, and this is actually really good at digesting carbohydrates. And starch digestion by amylase action anyways is virtually complete at the duodenal jejunal junction. So way up at the top of your small intestine, almost all of the carbohydrates digested. There's a lot of pancreatic amylase very active being secreted into the small intestine. This is a pretty famous enzyme. It was the first enzyme to be discovered back in the early 1800s. That's where the name comes from. It has amylone, starch in Latin. That'll help you remember. And it's a soluble enzyme that cleaves alpha-1 glycosidic bonds. And this is why amylase is going to help us digest our carbohydrates, right? Because we have these glucose linked by alpha-1 linkages. So its active site binds to 5 consecutive glucose molecules, and it cleaves between the second and third subsites. So it's cleaving in the middle of glucose polymers. So if you think of a long amylose, a long alpha-1 glucose, it's not chewing from the ends. It's cleaving all in the middle, breaking it into smaller pieces, right? So it's cleaving in the middle of these glucose polymers. So as I mentioned, there's a form secreted in the saliva. This doesn't seem to be regulated in any way. It just comes with the salivary flow. So salivary flow might increase during the cephalic response, and that will have a consequent increase in amylase. But amylase secretion, anyways, isn't really regulated at that level. In terms of the pancreas, though, it is regulated. So the pancreatic exocrine glands, and it's secreted into the pancreatic duct by secretods such as cholecystokinin or neural signals. And so these are the signals that are going to be inducing this fusion of the vesicle. Okay, so here's just a representation of what I meant by amylase binding to 5 glucose molecules. So here we have, you can imagine this to be a 1,4 linked, alpha-1,4 linked amylose. And amylase is going to bind to 5 glucose subunits, and it's going to cleave between that second and third subunit. So it's not cleaving from the ends, right in the middle. Okay, so for something like amylase, for amylose, if you expose it to amylase, eventually you'll end up with a lot of maltotriose and maltose. You'll end up with a lot of maltose, which is just a disaccharide, and a small oligosaccharide of glucose, right? So the kind of end products of amylase digestion of this long polymer will be 2 and 3 subunits of glucose polymerized together. Okay, and then there will be other enzymes that will be required to break these down further. For something like amylopectin or glycogen, in which it has those alpha-1,6 branch points, amylase can't break down the branch points. So in addition to maltotriose and maltose, you're also going to get these structures which are called alpha-limit dextrins. And these are molecules that have polymerized glucose through the alpha-1,4, and they also have that alpha-1,6 linked branch point. So if you expose amylopectin or glycogen to amylase, you get all 3 of these. You get maltotriose and maltose, and then also these alpha-limit dextrins. So it can't digest those any further. Okay, so in order to digest those further, as I mentioned, we can't absorb these. We need to break them down into single glucose monosaccharides in order for us to actually absorb those in our enterocytes. We need additional enzymes to break these remaining glycosidic bonds. And so this happens through the actions of brush-bordered dye and oligosaccharidases. And so these are going to be presented on the apical membrane of an enterocyte. So if we just imagine this is an enterocyte, so the apical membrane is facing towards the gut lumen, the basolateral membrane towards the blood plasma. We have ribosomes in the ER that are synthesizing these dye and oligosaccharidases. So these will be synthesized, they'll be subjected to glycosylation, which is our enzymatic addition of glycans, or sugar molecules, tracked through the Golgi, and then be finally transported via vesicular transport, snared air-mediated fusion with a plasma membrane presentation on the apical membrane. Okay, so this is a classic kind of secretory pathway, right? Okay, and so now we'll have these di and oligosaccharidases present on the brush border membrane. So this is in contrast to amylase, which was soluble. So those were just secreted into the duct, and they're going to be in the bulk phase lumen of the small intestine, moving around in that liquid. These are all stuck to the membrane. Okay? And we can see that here a little bit better in this representation of di and oligosaccharidases, in which we have one of these shown here. Now I'll walk you through the slide. It's a little bit busy. So here's the membrane, and it has what we call a transmembrane region, right? So it has a hydrophobic span anchoring into the membrane. So amylase is just going to be secreted, it's going to be soluble in the membrane spanning regions, whereas these guys are stuck in that brush border, okay, because of this transmembrane region. And then these different oligosaccharidases and disaccharidases have two active subunits. So shown here in this left corner of your slide is sucrase isomaltase. And so these are named for the primary activity of these two active sites. And so this is confusing because they have a lot of different nomenclature here, and that's because these have broad activity. So sucrase, which is most famous for cleaving sucrose, and this is primary activity, also has some maltase and glucoamylase activity. And that's why these are shown under here. Okay? So it's primary, think of it as sucrase. And then this one, isomaltase, so we see isomaltase listed here at the top, primary activity, but it also has some maltase and glucoamylase activity as well. So it's able to mediate these different effects, but the naming is based on its primary enzymatic role. Does that make sense? And so we have a few different brush border saccharinases. We have this one, sucrase isomaltase. And we'll talk about these in the context of alpha-limit dextrin digestion in the next slide. We have maltase glucoamylase. So maltase is going to break down maltose, which is two sugars, right? Two glucose sugars linked together. Glucoamylase cleaves glucose from the end of glucose polymer. And then we have lactase, making lactose. Fluorescein hydrolase, I won't really worry about that. That's another sort of uncommon activity present in the human. And then trehalase, I wouldn't really worry too much about this one either. This is a type of sugar present in shellfish, but it's not a huge contribution to our diet these days. So the most that we will be worrying about these two on the left, which are going to be mediating the breakdown of those glucose polymers and those starches that are the main carbohydrate in your diet. Okay, so this is a pretty good breakdown of the different enzymes that are present in this type of conformation and their substrates. Okay, so we have isomaltase. This is also called alpha-dextrinase, and this is going to mediate the cleavage of alpha-dextrins. So it actually mediates the cleavage of that alpha-1,6 branch point. Okay, so that's a really important one because we have to be able to cleave that branch point. Glucoamylase. It's also going to be cleaving dextrins, so it doesn't cleave the branch point. I'll show this on the next slide. We have maltase, which is cleaving maltose, sucrase, lactase, lactose, right? Okay, so let's take an alpha-limit dextrin through its remaining degradation. So if we had amylopectin or glycogen, it's been exposed to amylase, and broken down into this final alpha-limit dextrin, and amylase can't break this down any further. Right? And that's all happening in the bulk phase lumen of the GI tract. This then comes into contact with our saccharinases presented on that brush border membrane for further breakdown. So glucoamylase or maltase hydrolyzes alpha-1,4 link bonds and start clipping these sugars from the non-reducing terminus. Here we go. So we clip the sugar, and then we hit a 1,6 bond, which glucoamylase cannot hydrolyze first two bonds were 1,4 bonds. Right? Glucoamylase or maltase is hydrolyzing these, and now we've hit a 1,6 branch point. So this is where alpha dextrinase or isomaltase comes in to hydrolyze that 1,6 branch point. So here we have the branch. We hydrolyze it. Now we're down to just 3 glucoses molecules. Glucoamylase and maltase can again contribute to the breakdown. And finally, sucrase or maltase can break this final molecule down into free glucose subunits. Okay. And so let's have a little bit of overlapping activities, which can make this confusing. But one of the key ones I really want you to remember is this alpha dextrinase or isomaltase, which is cleaving those branch points. It's alpha 1,6 linkages. Basically, after amylase digestion, we have these alpha-lipodextrins. Glucoamylase or maltase is going to start cleaving back until it hits a branch point. This enzyme, alpha-dextrinase, comes in, breaks down that branch point, and then we can have further breakdown into free glucose subunits. And now we're able to absorb this. Okay, so in terms of the brush order diet in the liver saccharide regulation, their expression levels are increased carbohydrate ingestion, decrease of fasting. It's not always clear why this is, but it's probably due to insulin signaling. Okay, sucrase and lactase are very important disaccharidases. Lactase expression commonly declines as individuals approach adulthood. We'll talk a little bit more about lactase on a subsequent slide. And just recall and remember that the synthesis of the enzyme occurs within the enterocyte. This is in contrast to amylase, which comes from the pancreas. Okay, so brush-bordered disaccharidases and oligosaccharidases are coming from the enterocyte itself. Amylase from the pancreas. Okay, so just in terms of the activities, as I mentioned, sucrase is going to hydrolyze sucrose, which is glucose and fructose. That mediates hydrolysis and the release of these two free monosaccharides. This is just a different representation of what we've already discussed. And lactase is going to mediate the breakdown of lactose and glucose into their free monosaccharides for absorption. This is pretty easy to remember because their enzyme names are related to their substrates, right? Okay, so I think a lot of us are probably familiar with the idea of lactase deficiency. So, and there's also cases of sucrase deficiency as well. So lactase deficiency is more common. And this can result in indigestion. Okay? So if we're not having lactase breakdown of lactose in small intestines, so normally lactose is broken down by lactase, releasing glucose and lactose, which can be absorbed. That lactase is going to then traffic down to the large intestine and be available in the colon for microbial fermentation. Okay. This is why people with lactase deficiency, if they have dairy products with a lot of lactose in it, are going to have feelings of indigestion. This is because of microbial fermentation of that lactose in the large intestine, because we didn't break it down properly in the small intestines, and result in the production of some gases. Okay, and then very nicely, there are ways to treat this. If you do enjoy dairy products, lactate, and there's actually a comparable sucrate. And these are basically just enzyme supplements. Okay? So this supplies lactase to your body and allows you to break down lactose when you eat dairy products. If you have sucrase deficiency, you could also take sucrate and have sucrase and digest sucrase. So sucrase deficiency is common in infants because they're not getting much sucrase in the diet anyway. The main sugar they're getting is lactose from milk. Right? But it's rare in adults. So once an infant weans, the sucrase activity usually increases, and it's more rare in adults. Okay, but lactase deficiency is more common. So it affects most of the world's adults later in life after weaning. So here's just a kind of breakdown of how common this is. And there's one paper that's pretty interesting for this persistence of lactase activity found in a lot of European Americans. There's actually a mutation in an upstream coding region of this lactase gene, also referred to as the beta-galactosidase gene, that may be responsible for the continued expression of this enzyme. So maybe this mutation in the promoter region, which is responsible for lactase persistence in some individuals, whereas most individuals after weaning, after you're no longer getting milk from your mother, the lactase expression goes down. Okay, so in terms of carbohydrate digestion, I mentioned this earlier that it can be influenced by the physical and chemical characteristics of the rest of the food in the diet. So if you have a high amount of protein and fat intake, this is going to result in a slower carbohydrate absorption. There can be ingestion of amylase inhibitors. And so some of these occur naturally, and some of these have been developed specifically to slow digestion of sugars. And this is one way to help control kind of the spike diabetic patients get in their glucose after eating. So if you had take inhibitors of amylase or other enzymes that are breaking down the sugar, it slows the breakdown and slows the absorption and decreases that glucose spike after eating. So sometimes this is a good thing. So it'll reduce efficiency of carbohydrate absorption. And then, as I mentioned, also cooking and particle size can also influence carbohydrate digestion absorption, especially with that packing in the granules or those amylose hydrogen packing can make carbohydrates more resistant to hydrolysis by these enzymes. So cooking and getting those granules broken up can really assist your body with the enzymatic breakdown and hydrolysis of those bonds. In terms of indigestible carbohydrates, we'll talk about this more in the next class when we discuss fiber. But I just want to mention that this is because we don't have enzymes that can break down these specific linkages. So for cellulose, it is a glucose polymer. We use glucose in our body, but those are beta-1,4 linkages, and we just don't have enzymes that can break beta-1,4 linkages. Okay, but some animals do, right? Cows, other animals. Okay, I think we've gotten to the end. If you have any questions,